Welcome to our video lecture on topic 2.6, structure of DNA and RNA. The essential understanding for today, the structure of DNA allows for efficient storage of genetic information. That statement is also true for RNA. I'm not sure why we only put DNA in the understanding. Anyhow, our objectives, we are going to compare and contrast the nucleotides, those little pieces of DNA and RNA. We're going to do the same for strands of those nucleotides. We're going to describe this idea of anti-parallel structure when we're talking about DNA. We're going to look at Chargaff's rule for complementary base pairing. We're going to outline Rosalind Franklin's contributions to our understanding of DNA structure, as well as Watson and Crick's contributions. Our first objective is to compare and contrast the nucleotides, those little pieces of DNA and RNA. Remember that compare, we're looking for similarities, and contrast, we're looking for differences. We're both going to look at how nucleotides of DNA and RNA are the same, and then how they're different. One way that they are the same, they're all composed of three pieces: a sugar group, a phosphate group, and a nitrogenous base. We usually show the phosphate group with these circles that are attached to these pentagon shapes, these 5-carbon sugars, and then the nitrogenous base is going to have, you guessed it, nitrogen in it. The sugars, we do love to count the carbons when we're looking at biochemistry and organic chemistry. So carbon number 1 in the sugar is attached to the nitrogenous base. Carbon number 2, 3, 4, 5 kind of sticks up like this little flag. The 5th point of the pentagon is an oxygen, not the carbon. This little flag guy sticks up, and the phosphate group is stuck to carbon number 5, which is kind of nice because they both start with that F sound. So phosphate 5, they're stuck together. Same thing for DNA over here. So we have carbon number 1 with the nitrogenous base, 2, 3, 4, carbon number 5 up here in that flag thing, and then phosphate group on carbon number 5. Some differences between the two. The sugars are not the same sugars. The D in DNA is for deoxyribose. The R in RNA is just ribose. And the difference literally is we took off an oxygen, hence the deoxy. Notice here we have a hydroxyl group on carbon number 2 in the RNA sugar. And then in the DNA sugar, we took off that oxygen. We literally deoxied it. So there's just a hydrogen, no oxygen on that carbon number 2. Another difference is those nitrogenous bases. In RNA, we're going to have adenine, cytosine, guanine, and uracil. And then in DNA, we're going to have adenine, cytosine, guanine, and thymine instead of uracil. Otherwise, lots and lots of similarities. Let's also compare and contrast these chains of nucleotides, these polymers that we can make when we bind our nucleotides together. Both RNA and DNA have what we call a sugar-phosphate backbone. What that means is we're going to link the phosphates to the sugars to make this backbone of the molecules. So we're going to go phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down both RNA and DNA. Phosphate, sugar, phosphate, sugar, phosphate, sugar. So our sugar-phosphate backbones up and down all the strands of DNA and RNA. We're going to go ahead and count some of our carbons in the sugars, just to kind of look at what we're going to introduce, this idea of anti-parallel structure. So remember that in our sugar, carbon number 1 is stuck to the nitrogenous base, and we have carbon number 2 and 3 and 4. Carbon number 5 is up here next to the phosphate group. Again, carbon number 1, 2, 3, 4, carbon number 5, phosphate group. So when I have a phosphate group, which is stuck to carbon number 5, it's going to get stuck to carbon number 3 of the nucleotide that's next to it. Up here with the phosphate group, we're going to call it the 5' end because it's next to that number 5 carbon. Whereas down here, carbon number 1, 2, 3, 4, here's number 5 next to the phosphate group. The end of this guy we're going to call the 3' end because it's not next to the phosphate group. Same thing with our DNA. We're going to count our carbons. 1, 2, 3, 4, carbon number 5, phosphate group. This is going to be our 5' end. But look over on this side. Here's my phosphate group way down here. There's carbon number 5, and then I've got carbon number 1, 2, 3, 4, carbon number 5. 5' end is here. 5' end is here. If this is my 5' end, this is carbon number 3, here's my 3' end. If this is carbon number 5, then I've got carbon number 1, 2, 3, 4, carbon number 5 next to the phosphate group. Carbon number 3 is here. Notice that they're going opposite each other. So I have on one side 5 and on the other side 3. Over here I've got carbon number 5. Over here I have carbon number 3. And this is what we call anti-parallel. This idea, anti-parallel strands of DNA, we're going in opposite directions. So I have 5' carbon. Carbon number 5 is up here. I have carbon number 5 down here. And so they're going to be on opposite ends. We're still building these chains of nucleotides, but they go in terms of the carbon numbers in the sugars, they go in opposite directions, hence anti-parallel strands. And then those anti-parallel strands are going to be held together by hydrogen bonds between the complementary bases. So I have adenine here and thymine here. Adenine and thymine, the structure of them is just right that they can build these hydrogen bonds to hold them together. Same thing between guanine and cytosine. I've got guanine here, cytosine here. Their structure is just right that they line up and build these beautiful hydrogen bonds. Remember hydrogen bonds from when we talked about water. Hydrogen bonds are forces of attraction, so not like an actual chemical bond, but almost like a little magnet. These forces of attraction that hold molecules together when they're quite polar. So we'll have the oxygen here and the hydrogen here, and they just kind of get stuck together like little magnets. I'm going to repeat a lot of the words from the previous slide, zoomed in on the picture because it's way easier to see when the picture is big. So again, carbon number 1, 2, 3, 4, 5 phosphate groups stuck to carbon number 5 makes this the 5' end of this strand of DNA. At the other side, I have carbon number 1, 2, 3, 4, 5. There's my phosphate group. And so this is what we call the 3' end because there isn't a phosphate stuck to carbon number 5 at this side. This is the 3' end. On the complementary side, the other piece of this double-stranded DNA, notice that I've got my phosphate group here, which means that this is carbon number 5, which means that this is the 5' end. Here was 3, here is 5. And then at the opposite side of this strand, all the way up here, I have carbon number 1, 2, 3, 4, carbon number 5 next to the phosphate group. This guy ends in carbon number 3. This is my 3' end, but over here it was 5, but over here it is 3. 3 to 5, 3 to 5 in opposite directions. This is anti-parallel. When we start talking about DNA replication and transcription, that's how we take the DNA to make messenger RNA. When we talk about translation, which is how we take the messenger RNA to make a protein, these directions, 3' to 5', 5' to 3' is going to be super, super important. So start thinking about it now, put it into the back of your brain, let it ruminate for a little bit. When we get to our metabolism unit, this is going to come back big. I'm going to throw in a little bit more detail about our nitrogenous bases here. You may have noticed that adenine is a little bit bigger than thymine. Guanine is a little bit bigger than cytosine. Adenine and guanine are what we call purines, and they have these 2 rings. Adenine and guanine are purines. And then the other guys, the smaller guys, are pyrimidines. So cytosine and thymine, I'm going to throw uracil in there as well. Uracil is also a pyrimidine with only 1 ring. What's lovely about how these guys pair is that we have one of the bigger guys and one of the smaller guys in every pairing. A big guy and a small guy, a big guy and a small guy. And this allows for the DNA to have just the right width. We don't want to try to smash 2 guys that have 2 rings next to each other because then we're going to have like, imagine I had a whole other ring, it's going to like bulge out and then it won't be lovely and smooth. It'll be hard for our enzymes to get to the DNA to do all the work that they need to do. So this lovely matching of one slightly larger, one slightly smaller, one purine and one pyrimidine helps just to keep the DNA nice and smooth, a nice width. How to remember what the purines are and what the pyrimidines are. I do have a little mnemonic device that you might want to try. It doesn't make a whole lot of sense, but it works for me. It may also work for you. So I remember 2 puga Why cut one? 2 puga? Why cut one? And you're like, what does that even mean? Well, our purines have 2 rings. Guanine and adenine are our 2 double-ringed purines. Y for pyrimidine, cytosine, uracil, thymine, 1, they only have 1 ring, right? So 2 puga, purine, guanine, adenine, 2 rings. Why cut one? Y from the pyrimidine, cytosine, uracil, thymine, 1 ring. Oh, maybe no, I don't know. It works for me. It doesn't work for other people, but you can try it. And then one other little piece that I'm going to throw in, one more tiny little detail. So there are 2 hydrogen bonds that fit the adenine and thymines together just right. There are 3 hydrogen bonds that hold guanine and cytosine together. This one I think is easier because G and C and 3, they rhyme. 3 hydrogen bonds, 1, 2, 3, between guanine and cytosine, GC3. Whereas A and T is only 2, AT2. It kind of works. Good. Purines, pyrimidines, 2 puga. Why cut one? AT2, GC3. You've got all the details. Good? All right. So let's talk about how we figured out that adenine and thymine pair together and cytosine and guanine pair together. How do we know this? This super guy, Edwin Chargaff, helped us figure this out. So he took lots and lots of tissues from lots and lots of animals and then just kind of did some analysis. And he figured out that in the thymus of an ox, that most of the time we had fairly equal numbers of adenine and thymine. And we had fairly equal numbers of, and again, they're not perfectly equal, but they're pretty close, of cytosine and guanine. Same thing when he looked at man, pretty equal-ish numbers of adenine and thymine, equal numbers of guanine and cytosine. This just led him to figure out, must be that A's and T's pair together, C's and G's pair together, because we had those equal-ish percentages in all these different tissue types. We're going to throw one more little piece of science history into our story for the day, and then we're done with this lecture, my friends. We've been saying that DNA is a double helix. What that means is it's double-stranded, and then it kind of twists, it spirals a little bit. Double helix. How did we figure that out? Let's talk about the story of Rosalind Franklin, James Watson, and Francis Crick. So very similar to electron microscopy, where we shoot electrons at a sample and then the electrons bounce off and then the computer collects that information and then we can figure out what stuff looks like, our electron microscopes. Very similar to that, we have X-ray crystallography. In X-ray crystallography, we crystallize our sample, shoot some X-rays at it. How those X-rays bounce off the crystallized sample produces what we call a diffraction pattern. We can analyze that diffraction pattern, do lots of calculations to kind of work backwards and figure out what the molecule must have looked like in order to produce that diffraction pattern. Rosalind Franklin was an X-ray crystallographer, and she took an amazing picture of DNA. Here is that amazing image. It was actually taken by Raymond Gosling, a grad student in Franklin's lab. Franklin did the work to analyze, interpret, do the calculations, the measurements, so that it could actually be used by these guys to figure out what the shape of DNA was. Rosalind Franklin did the calculations. The image was taken in her lab. These guys used her interpretations to deduce that DNA must have been a double helix shape. These guys won a Nobel Prize for that work. Rosalind Franklin was not even mentioned. So it's an interesting story. Is it okay that Rosalind Franklin wasn't acknowledged because science is science and knowledge is power and it's just important that we learn things? Or should she have been recognized for her contributions? It's a big question. It's a big question that we ask in science very frequently because the people who do the work are frequently not recognized for it. And on that note, we have completed our video lecture on the structure of DNA and RNA. We know that the structure of DNA, these sequences of nucleotides, allows for the efficient storage of genetic information. When we get to our unit on genetics, we will talk about how we can use these sequences to code for some messenger RNA, to code for some proteins. Super important. We accomplished our objectives. We compared and contrasted the nucleotides of DNA and RNA. Both of them have phosphates and sugars and nitrogenous bases, but those nitrogenous bases are different and the sugars are different. We looked at the strands of DNA and RNA. Both have sugar phosphate backbones, but DNA is double-stranded and RNA is most of the time single-stranded. We talked about anti-parallel structure. Remember that we have the 5' carbon next to the phosphate group. 3' carbon is the other end. But when we're looking at our double-stranded DNA, we have 3' next to the 5' and 5' next to the 3'. Trogoff was the guy who figured out that A's and T's go together and C's and G's go together. Rosalind Franklin took that amazing picture that was used without permission by Watson and Crick, and that's how we know that DNA is a double helix. Good work today.